Welcome to an exploration of one of the most fascinating frontiers in computing, liquid processors that compute with chemistry instead of electrons. Imagine a world where computation happens not in silicon chips, but in tiny droplets of liquid, where chemical reactions replace electronic circuits. Let's start with a fundamental question. What exactly is chemical computing, and why should we care? Traditional computers work by controlling the flow of electrons through silicon transistors, switching between on and off states. Chemical computers instead use molecular reactions as the basis for information processing, where different chemical states represent different computational values. The key difference: silicon computers manipulate electrons, while chemical computers manipulate molecules and their reactions. Now let's dive into the heart of our topic, the microdroplet system. Microdroplets are tiny liquid spheres, typically ranging from 1 to 100 micrometers in diameter, about the size of a human cell or smaller. A typical droplet might be 10 micrometers in diameter, or 10 to the power of -5 meters. Inside each droplet, we have different chemical species that can react with each other, creating a dynamic computational environment. Let's calculate. What's the volume of a 10 micrometer diameter droplet? Using the sphere volume formula, we get 4/3 pi times r cubed, where r is 5 times 10 to the -6 meters. This gives us approximately 5.2 times 10 to the -16 cubic meters, or 520 femtoliters, an incredibly tiny volume. Here's where it gets exciting. How do we build logic gates using chemistry? Logic gates are fundamental building blocks of computation, performing simple operations like AND, OR, and NOT on input signals. In electronic circuits, these gates control electron flow, but in chemical systems, we use reactions. Let's design a chemical AND gate using a real example. In a chemical AND gate, product C forms only when both reactants A and B are present simultaneously. Input A and input B enter the reaction chamber, and output C emerges only when both inputs are present, just like a digital AND gate. Let's explore a real chemical system that exhibits computational behavior, the Belousov-Zhabotinsky reaction. The Belousov-Zhabotinsky reaction, discovered in the 1950s, is an oscillating chemical system that cycles between different states periodically. The simplified reaction involves cerium ions, bromate, and malonic acid, cycling between cerium 3+ and cerium 4+ states. The concentration of cerium 4+ oscillates periodically over time, creating a chemical clock that can be used for computation. The oscillation period typically ranges from 10 to 60 seconds, much slower than electronic circuits but useful for certain applications. An important question: how fast can chemical computers actually compute compared to silicon? Chemical computing speed is fundamentally limited by how fast molecules can diffuse and how quickly reactions occur. Diffusion time equals L²/d, where L is the distance and d is the diffusion coefficient. Let's calculate for a 10 micrometer droplet using typical water diffusion values. Water molecules have a diffusion coefficient of about 10 to the -9 square meters per second. Plugging in our values, diffusion time equals 10 to the -10 divided by 10 to the -9, giving us 0.1 seconds. Chemical computing operates on millisecond to second timescales, vastly slower than gigahertz electronic processors. Electronic computers work at nanosecond timescales, making them roughly 1 billion times faster than chemical systems. This 9 order of magnitude speed difference is the major challenge for chemical computing. But here's the twist. Chemical computers have a superpower that silicon lacks. Silicon processors work serially, executing one operation at a time per core, even with multiple cores. Chemical systems offer massive parallelism, where millions of droplets can compute simultaneously in parallel. Serial processing handles tasks sequentially like a single-file line, while parallel processing handles many tasks simultaneously like multiple checkout lanes. How do we actually encode information in chemical droplets? There are multiple ways to encode information in chemical systems, each with advantages. Concentration encoding uses the amount of a chemical to represent information, like high concentration equals 1, low equals 0. If concentration of A exceeds a threshold, we call it 1, Below the threshold, we call it zero. Typical thresholds range from millimolar to molar concentrations, depending on the specific chemistry. Color encoding produces visual outputs, where different colors represent different computational states, making results easy to read. Red droplets might represent binary 0, while blue droplets represent binary 1, creating a visible computational output. Can we combine these chemical gates to build more complex circuits? We can cascade reactions where the output of one gate becomes the input of the next, building complexity. 3 gates in series create a processing pipeline, where chemical signals flow from input through multiple processing stages to output. Major challenges include signal degradation as we cascade more gates, and keeping timing synchronized across parallel pathways. Let's examine the energy requirements of chemical computing. A critical question: how much energy does each chemical computation require? The energy comes from Gibbs free energy, which equals enthalpy change minus temperature times entropy change. Typical chemical reactions release 10 to 100 kilojoules per mole of product formed. Per molecule, this equals about 10 to the negative 19 joules, obtained by dividing by Avogadro's number. Modern transistors use about 10 to the negative 18 joules per switching operation, roughly 10 times more than a single molecular reaction. However, each droplet operation involves billions of molecules, not single molecules, changing the comparison. A droplet computation requires about a billion molecules times 10 to the negative 19 joules, giving 10 to the negative 10 joules. So chemical operations use more energy per gate, but the massive parallelism can compensate for certain problem types. Where would liquid processors actually be useful? Biomedical diagnostics could use droplet computers to detect disease markers, performing complex analysis on tiny blood samples. Blood enters the system, chemical logic gates analyze the markers, and results appear as color changes or fluorescence. Environmental sensing applications could deploy droplet networks to continuously monitor water quality and detect pollutants. Drug screening could test thousands of chemical combinations simultaneously in parallel droplet arrays, accelerating discovery. Chemical synthesis planning could use liquid processors to explore reaction pathways and optimize synthetic routes. What are the major obstacles preventing widespread adoption? Reproducibility is challenging because even tiny variations in droplet size or composition can alter computational behavior. Scaling up requires controlling millions of droplets simultaneously with precise timing and positioning. Programming chemical computers is difficult because we lack universal chemical programming languages and standardized gate libraries. Integration requires interfacing chemical systems with electronic controllers, bridging vastly different time and energy scales. 3 major technical hurdles: precise fabrication of droplet arrays, real-time control systems, and reliable readout mechanisms. Microfluidics provides the physical platform for manipulating droplets. Microfluidic channels are microscopic pipes, typically 10 to 100 micrometers wide, that control fluid flow with extreme precision. Flow behavior is characterized by Reynolds number, which equals density times velocity times length divided by viscosity. In microfluidic systems, Reynolds numbers are typically much less than 1, meaning flow is laminar and predictable. With typical values, Reynolds number equals about 0.1, confirming smooth laminar flow without turbulence. Droplets flow smoothly through microfluidic channels in single file, with precise control over spacing and timing. How do we actually create these computational droplets? T-Junction geometry creates droplets when 2 immiscible fluids meet at perpendicular channels, forming regular-sized drops. Flow focusing uses surrounding flows to squeeze a central fluid stream, breaking it into uniform droplets. The continuous phase flows horizontally, while the dispersed phase flows vertically, meeting at the junction to form droplets. Modern microfluidic systems can generate between 100 and 10,000 droplets per second with high uniformity. Droplet generation frequency equals the flow rate Q divided by individual droplet volume. How do droplets communicate to form computational networks? Droplet fusion allows 2 droplets to merge, mixing their chemical contents and combining computational signals. Droplet fission splits one droplet into multiple daughter droplets, distributing signals to parallel pathways. 2 input droplets approach and fuse into a single, larger droplet containing the combined chemical signals. Droplets can also communicate by proximity, where small molecules diffuse through the separating medium between nearby droplets. Let's examine a cutting-edge application: DNA computing inside droplets. DNA strand displacement reactions can perform logical operations where DNA strands competitively bind and release each other. In strand displacement, input strand A displaces strand C from complex BC, forming new complex AB and releasing output C. Each microdroplet serves as an isolated reaction chamber where DNA logic gates operate independently. Inside the droplet, DNA strands bind and unbind according to complementary base pairing rules, implementing logical functions. DNA computing offers highly specific molecular recognition through Watson-Crick base pairing, enabling complex circuit design. The main limitation is slow kinetics, with complex DNA computations taking hours to complete. Let's directly compare different computing approaches. Serial processing handles tasks sequentially like a single-file line, while parallel processing handles many tasks simultaneously like multiple checkout lanes.